for giving me this opportunity to be able to present my work. And yes, just to reiterate this point, please do forgive me for any mistakes, as I was able to turn my poster into a PowerPoint presentation in the last 5 minutes. I'm going into my second year of my master's with the Center for Neuroscience, or Neuroscience and Mental Health Institute at the University of Alberta. And this will be my last year as a master's student. And my project is on focusing to characterize the mode of action of beta-2 adrenergic receptor agonists, a possible therapeutic for Parkinson's disease. So why am I studying beta-2 adrenergic receptor agonists as a potential therapeutic for Parkinson's disease? So as you all know in the many presentations that have been going on today and yesterday, that Parkinson's disease is a debilitating disorder, and we really need to work together to be able to find a cure or even find some way to be able to manage the disorder. And previous work from our laboratory has shown that salmeterol, which is a beta-2 adrenergic receptor agonist, to be highly effective in inhibiting the progression and actually reversing the neurodegeneration that happens in a Parkinson's disease, but in the animal model. However, right now we have a commercial availability of the super long-acting beta-adrenergic receptor agonists, vilantrol and indicaterol, and because they are super long-acting, you would think that they would be acting better at the synapse or at the beta-2 adrenergic receptor as an agonist. And so my goal is to be able to characterize exactly how these two super long-acting beta-adrenergic receptors work and compare them to that of salmeterol, because salmeterol has already been shown to be effective in reducing or inhibiting the progression of neurodegeneration in an animal model of Parkinson's disease. So if I do find that it is similar to that of Parkinson's disease, then it makes the work much more easier in being able to take it to the next level. And so I had 3 specific goals in this project. The first one was to determine the effective dose and kinetic of action of vilantrol and indicaterol and compare that to salmeterol, and determine the effects of vilantrol and indicaterol on the phenotype and function of microglial cells, because keeping in mind that within the CNS, despite the fact that there's a lot of, maybe not despite, but in collaboration with the many other cells that are present in the CNS, microglia is a major player as an inflammatory cell. And so I wanted to be able to characterize this function in microglial cells, or at least the anti-inflammatory effects of it in microglial cells. And lastly, my goal was to determine which intracellular signaling molecules are activated or inhibited by these beta-2 adrenergic receptor, because if we can figure out the signaling cascade of how these molecules work, then it makes it easier for us to be able to administer it into humans, knowing how they function and being able to control possible side effects if we know how these things work. So as part of my methods, what I do is I work with the BV2 murine microglial cells for, focusing in on the anti-inflammatory effects of these drugs on microglial cells. And these microglial cells, they're kept in standard cellular conditions, like the 5% CO2 at 37 degrees, just like any other cell culture would be kept. And with all of my work, what I do is I seed the microglial cells the day before, because I want to be able to make sure that these cells are not agitated when I give them the drug, to be able to reduce confounding their variables. And for my kinetic studies, I do pre-treatment the night before, and then the next day I do pre-treatment with the beta-2AR agonist at the various concentrations for 45 minutes, and then I take it out and add in fresh media with LPS for 4 hours. And I've chosen 4 hours because I've done kinetic studies for 1 hour, 2 hour, 4 hour, and 24 hours, and I found that 4 hours to be a good timing to be able to allow the inflammatory cytokines to be released into the supernane. And the kinetic study is to be able to figure out the effective dose of these drugs. And the inhibition study is to be able to figure out my third point, which is which intracellular molecules are activated or inhibited by these beta-2 adrenergic receptor agonists. And for these, I did pre-treatment with the inhibitor for 30 minutes, then I did my beta-2AR agonist pre-treatment, then I did the inflammagen or the LPS treatment, because LPS is supposed to activate microglial cells. And so going back to my objectives, the first objective and the second objective being able to determine the effective dose and kinetics and to be able to determine the effects of these beta-2AR agonists on the function and phenotype of microglial cells. So first of all, I chose two of the pro-inflammatory cytokines to focus in on, TNF-alpha and IL-6. And with TNF-alpha, what you see is that when you do a beta-2AR stimulation, it actually inhibits the level of TNF that is produced. Sorry about here. Sorry about this. I don't know what happened when I was transferring, but what it is that the first line is BV2 cells, the second is LPS, and from here to here is the concentration gradient, and this would be for Vilantrol. So this is when you pre-treat the cells, the BV2 cells with Vilantrol for 45 minutes, and then you do a post-treatment with LPS for 4 hours. You have a spike. When you have an LPS only, you have a spike of like 800 picograms of TNF-alpha. However, when you have it pre-treated with Vilantrol, you have a significant decrease, almost comparative to BV2 cells without any treatment. So that's pretty good. That's pretty promising. And the story is the same in terms of Indicadrol and in terms of Salmeterol. And what you see is that it's pretty effective at all doses, all the way up until 10 to the negative 10th molar. That's pretty good at these levels. And then in terms of IL-6, the story is not what you expected, because I expected the same as that I would see in TNF, because they're both pro-inflammatory cytokines. So I thought that, for a drug to be anti-inflammatory, it should reduce levels of both TNF and IL-6. But what I found was that TNF was reduced. However, IL-6, this is LPS, and these are the various concentration of the drugs. So Salmeterol is here, Indicadrol, and this is supposed to be Vilantrol. And what you see is that IL-6 is either enhanced or actually the same level as BV2 cells, and these are all significant in comparison to BV2 cells. So the level of IL-6 is actually not inhibited, but rather either enhanced or the same level that you would get when you just give them an inflammagen LPS. So that was really interesting for me to see and for our lab to see. So now going into my third objective, which is to determine which intracellular molecules are activated or inhibited. So first of all, to be able to establish that these beta-2AR agonists really do work through the beta-2AR receptor, I did a study with to inhibit the beta-2AR receptor with propranolol. And what you find is that with TNF, you have the LPS treated cells, and you have like a significant increase in the level of TNF. When you do a pre-treatment with Indicadrol, and I chose one of the drugs to do it because, as you can see, that the number of graphs would be significantly overwhelming. When you do a pre-treatment with Indicadrol, you have a significant reduction in the level of TNF-alpha. However, when you block the beta-2AR, the level goes back up, so that's pretty good. So you know that these beta-2AR receptor agonists actually do function through the beta-2AR receptors. However, the story with the IL-6 is that Indicadrol, as I had shown previously, has no effect on the level of IL-6 in terms of inhibiting it. And when you do an antagonist to beta-2AR, obviously, because it doesn't function through the beta-AR, there is no effect. So that was also pretty good. And the next thing I wanted to see was whether these beta-2AR agonists would have any effect through the major inflammatory pathway, the NF-kappa B pathway. And so what you do with the inhibition studies, or what I did, was I gave them compound A, which is an inhibitor of the NF-kappa B pathway. And as you can see, that when you inhibit NF-kappa B, now going back, so it is well known that TNF is produced through the NF-kappa B pathway. IL-6, there's a lot of literature and a lot of understanding that IL-6 is also controlled through the NF-kappa B pathway. However, there's also literature that says, IL-6 is also regulated by other pathways. So what I found was that when you inhibit NF-kappa B, that actually the level you would assume that when you give a NF-kappa B inhibitor that it should rescue the level of TNF-alpha. However, you don't see that. And that Salmeterol, it inhibits it, and compound A, an inhibitor of NF-kappa B, has no effect on either rescuing it or further inhibiting it. IL-6, however, what you see is that in the presence of the beta-2AR agonist, it's either enhanced or, at the same level as the LPS, which is consistent with my previous result. However, when you give a NF-kappa B inhibitor, you have complete abolition of the IL-6 response. So now this tells me that the beta-2ARs, they activate the NF-kappa B pathway in terms of stimulating the release of IL-6. The other molecule that I was interested in is called TAC1, or also, it has a variety of names, but one of the major names that it's known by is the transforming growth factor beta-1. And it is one of the cytokines, it's a major cytokine that's responsible for cell proliferation, apoptosis, and growth and differentiation. So that's why I was interested in this, because with Parkinson's disease and cell death, you would want to know what the major molecules that are responsible for cell proliferation or apoptosis, which one of these would be involved in any particular drug that you prescribe Parkinson's patients. And when you do give an inhibitor to TAC1, known as 5C7-oxazenol, at all concentration, both with TNF-alpha and IL-6, it completely inhibits the levels of TNF-alpha and IL-6. So now this is pretty good data that says that the beta-2 adrenergic receptors function in terms of either causing the release of IL-6 or inhibiting the release of TNF-alpha. Both of these effects of the beta-2AR function through the TAC1. And P38 is another major cytokine, just like TAC1, in terms of controlling cell proliferation, apoptosis, differentiation. And so I looked at this one as well. And when you give an inhibitor to P38, there isn't any significant difference between in the TNF level or the IL-6. So this tells me that the beta-2AR agonists function independently of the P38 signaling molecule. As you can see that the levels are pretty consistent throughout, whether you give an inhibitor or not an inhibitor. And same thing with salmeterol. Despite the fact that you see a decrease, it's not significant. And the other particular molecule is JNK. And JNK is also one of the major signaling molecules that is responsible for controlling the fate of a cell. And what you see is when you give a JNK inhibitor, SP600-125, I always asked my professor, I'm like, why would they come up with these kind of names when they could come up with almost a much more interesting name, but this is what we get stuck with. When you give them the JNK inhibitor, you can see that the level of TNF-alpha is inhibited furthermore, and IL-6 is inhibited. So this tells me that the JNK functions with the beta-2AR in a synergistic manner in terms of inhibiting TNF, and the fact that the beta-2ARs didn't inhibit the level of IL-6, JNK comes in to actually inhibit those levels. So I know that JNK plays a major role in terms of this particular signaling pathway. And so to summarize, these beta-2AR agonists, they're promising anti-inflammatory therapeutic. However, we can't get carried away, and we need to be able to really do the groundwork in understanding how they function, because as I have shown that, it may have a good effect in terms of one pro-inflammatory cytokine, but it may have a significantly different or no effect on another cytokine. And you would want to be able to put out on the market a drug that works the best for Parkinson's patients, because you don't want to give them one thing that only works halfway through. Because despite the fact that I have shown that IL-6 is not affected by beta-2AR or in fact enhanced, there's a plethora of other cytokines that are good and bad that I haven't even looked at. So it's something that you need to be very rigorous at when you're trying to take a drug to clinical trials or to be able to extend it to patient care. And so to summarize, I guess my points is that the beta-2AR significantly inhibits the LPS-mediated TNF, but it has no effect and does not inhibit the LPS-mediated IL-6 response. And the effect on both TNF and IL-6 are P38 and PKA. I actually had to take this out because I wanted to make my presentation short, but it's on my poster if you want to look at the data for that. And the inhibition of NF-kappa B with compound A inhibits the IL-6 and does not rescue or enhance the TNF response. And that the beta-2AR inhibitory effect may be mediated through the JNK pathway. So in conclusion, characterizing and understanding the mode of action of beta-2AR agonists can provide significant insight into how these drugs possibly can work to reverse CNS inflammation, and may provide a more effective therapeutic for Parkinson's disease once you've able to carve out the detail in terms of all of the pro and anti-inflammatory cytokines that are present in the Parkinson's brain. And thanks to all of these funding agencies that have been kind enough to support our work. And I didn't mention it, but thanks also to my professor, Dr. Patrick Flood, who has really guided me through this project, because I can tell you it's very, very complex. So thank you for your time.